Okay, so hello and welcome back to Excelsior Academy. So in the previous lecture, we have discussed about the CDKs and the cyclins, like how they are regulating the G1 phase, S phase, G2 and M, right? Now in this lecture, it is basics, that is the mitosis stages. So already this we know. In this lecture, we will just go through all the different stages of the mitosis, like prophase, metaphase, anaphase, and telophase. Okay. And we also know that after mitosis, the cytokinesis takes place, which results in the formation of the two daughter cells and those two daughter cells will be in the G1 phase. Okay, so this is like very basic, which we have learned. So I'll be just going through all those the content. If anything, any doubt is there, guys, you can ask. Okay, this is like very basic. Mainly the exam with the exam point of view, the question will not come because ये हमने बहुत पहले से पढ़ते आ रहे हैं, like how what happens during the prophase or metaphase or anaphase. थोड़ा सा anaphase is more important because there so many checkpoints are there. Okay, if you remember, we have talked about the APC, anaphase promoting complex. So there are two types are there, 1 and 2. So that is something which is important. Okay, rest all phases, like we can just go through it, the content. Okay, so it is the period between the two mitotic stages. The chromosomes are decondensed and dispersed throughout the nucleus, and it is in the form of the chromatin. Okay, now questions कैसे आ सकते हैं, like this is for the interview ways. Okay, अगर chromatin case में है, so when you say chromatin, then what all proteins are present? So you can talk about the histones over here. Okay, then you can talk about HAT and HDAC, like ये कैसे काम करते हैं whenever you need any particular region of DNA to be transcribed, like for the gene expression. Okay, so आप ऐसे discuss कर सकते हो. So mainly in the interface, it is in the form of the chromatin. So the shape of the cell appears uniform. The first stage of interface is G1 or the growth stage 1, which determines the fate of the cell. यानी कि क्या? Either it will go for the differentiation or it will enter the G0. ठीक है, so we talked about the G0 stage, which is also known as the quiescent state, and it is reversible. So we know that when a cell is there and you don't have proper growth factor or the nutrition available, okay, then in that case the cell enters the G0. अब इसमें भी एक चीज आपको समझना है, G0 of a animal cell depends on the growth factor, whereas for the bacterial cell it depends on the nutrition. ठीक है? Then it proceeds into the S phase, comes to complete the cycle of the division. Duplication of genome happens in the S phase. The second gap phase is the last period preceding the initiation of the mitotic prophase. Okay, and duration of these phases vary in different kinds of the cells. So this already we have discussed. So basically, mitosis will take around 1 hour. G1 is around 11, S is around 8, G2 is around 4. Okay. 12, 24 हो गए. Yes, correct. Now, during interphase, the cell is prepared either for the mitosis or the meiosis. Somatic cell will undergo mitosis to reproduce themselves, while the germ cell will undergo meiosis to produce the haploid cells for the purpose of sexual reproduction. Okay, so if you see in the figure also, you have got the two centrosomes, plasma membrane, nuclear envelope is there, chromatin nucleus is present. Okay. So this lecture is going to be very basic, guys, because हमने ये बहुत पहले से पढ़ा हुआ है. ठीक है, प्रोफेज क्या है, मेटाफेज क्या है. Now, in early prophase, the chromatin fiber starts getting condensed to chromosome. Nuclear membrane degradation takes place. Now, what are the proteins which helps in the condensation of the chromosomes? We discussed in the last lecture. Tanvi? Shruti? Proteins responsible for condensation of a chromosome?
Condensation.
And one more protein. So those are your condensins and cohesins, right? Okay. Then the nuclear envelope of the membrane degradation takes place. So this also you know that is because of the phosphorylation of your lamins. Okay. Now, lamins protein gets free from the inner nuclear lamina to form the protein scaffold onto which chromatin fiber loop out to form the chromosome. Now, the vesicles are generated due to nuclear membrane degradation remain present in the cytoplasm. So, जो आपके nuclear membrane जो disappear हुए हैं, so that is like वो छोटे छोटे से आपके membrane fragments हो गए हैं like this. Okay, and they remain near to your chromosomes जहाँ पे भी आपके chromosomes present है. So what happens is that when they get separated, like here, when they get separated, जो chromosomes के पास ऐसे nuclear membrane है present like this, they come together and fuse. So slowly they reappear. Okay. Microtubule formation restores in the prophase. Centrosome gets split to form the two centrioles, which moves towards the opposite pole, and acid rays formation from the centriole gets started. Thus, in the late prophase, spindle apparatus formation gets started. Okay. Late prophase, microtubules begins to search for the kinetochore. Kinetochore is a motor protein attached at the centromere of a chromosome, and it is a point of attachment site to the microtubules. Now that is a place where all these motor proteins will come together. They will bind to the chromosome, and once the sister chromatid gets separated, they will take the single sister chromatid towards the opposite pole, like walking on the microtubules. Okay. Now coming to the metaphase. So in metaphase, centriole reach out at the opposite pole and spindle apparatus gets established. So we know that in the case of metaphase क्या होता है, आपके सारे chromosomes जो हैं एक single plate में आ जाते हैं, ठीक है? So at the pole, centriole give rise to three types of microtubules. So we know those are your astral microtubules, kinetochore, and polar. Okay. So astral microtubule which remains in the cell cortex. It also gives rise to kinetochore microtubule which gets attached to the chromosome at the kinetochore through search and capture mechanism and arranges them with the spindle apparatus at the metaphase. Okay. The another one is the polar microtubule. The two centrosomes begin pulling the chromosome towards the opposite end of the cell. The resulting tension arranges the chromosome to metaphase place. It is the third type of microtubule, that is the polar microtubule. From both sides overlap each other and help in the chromosome movement. So this also we have discussed in the previous lecture, like how astral and the polar microtubules are helping in the separation. I hope everyone remembers. Now coming to the anaphase. So anaphase mein kya hoga? The sister chromatids will get separated. Okay. Now each sister chromatid, now called chromosome, will move towards the opposite side. Now this is done by the two forces, poleward force and the polar ejection forces. In the case of the late anaphase, chromosomes reaches the opposite pole, and early anaphase is known as the anaphase A and the late one is known as the anaphase B. We'll come to the anaphases A and B. Also, we have discussed earlier also, like how it is behaving. Now, telophase में क्या होगा? Almost this cleavage furrow will form and the contractile ring will come. So, contractile ring क्या है? It is again nothing but the formation of the ring by actin filaments. Okay. And then it will lead to the cytokinesis. So where two cells are there, as you can see that the nuclear envelope starts reforming and the chromosomes will start decondensing. Okay, it will start becoming the chromatin. Now cytokinesis. So in animal cell cleavage, all cleavage furrow forms by the actin and myosin 2, which is a protein, motor protein, derives the cytokinesis process. So this also we have discussed already, actin and myosin 2, how they are helping in the cytokinesis. Now who can tell me the action of actin plus myosin 1? ये हमने कहाँ देखा है? In which case, which type of movement we have seen the action of actin and myosin 1? Yes, very good. Microvilli. Good. So mitosis is complete with the following the cytokinesis. In the plant cell, a cell plate is formed in the middle of the cell. This is made up of vesicle derived from the Golgi apparatus. It is important part for the maintenance of the chromosome set. Mitosis helps in the growth and development. A single zygote is converted into multicellular body through mitosis. Tissue regeneration is also achieved by mitosis. Asexual reproduction like budding in hydra or in any lower animal is a type of mitosis only. Okay, again, vegetative propagation in plant is also mitosis. Now, a mitotic error can create aneuploid cells having too few or many one of the chromosomes. So it will result in the cancer. So what we are saying is that during mitosis, if there is no equal separation of the chromosomes, then it will result in the aneuploidy. यानी कि क्या होगा कि किसी केस में sister chromatids like this they are present, right? So instead of going in the opposite direction, sometimes both go in the one direction. तो क्या होगा एक सेल में लेस नंबर ऑफ क्रोमोसोम्स रहेंगे और दूसरे सेल में आपके पास ज्यादा रहेंगे. ठीक है, and that's how the aneuploidy starts. Now coming to the meiosis. So it is a specialized type of cell division. It reduces the chromosome number by half, creating 4 haploid cells. It is for the sexual reproducing cell. Now in meiosis, DNA replication is followed by 2 rounds of cell division, and the meiosis is divided into 2 categories, 1 and 2. So interphase of meiosis. Nuclear material gets double in this place, and central replication also takes place in the interphase. Here the vesicle fuse with each other at the center of the phragmoplast and develops into a cell plate in the case of the plant. Okay. Now in the case of interphase, again 3 phases are there. So this we know, G1, S and G2. I'm not repeating. Okay. Meiosis 1. So this is a reduction division. Meiosis 1 is segregation of homologous chromosome. Meiosis 1 produces haploid cell. Okay, so in which it contains N or the 2N. Thus the ploid is reduced from diploid to haploid, and uske baad jo hota hai, that is similar to your mitosis. That is meiosis 2. Everyone knows, right? So thus meiosis 1 is called the reduction division. Prophase 1, it has got 5 stages. Now, in the case of meiosis, this prophase 1 is also important. There are different stages, right? So let's discuss all these. Guys, you can stop me in between, okay? If you have any doubt, ये बहुत basic है, so that's why I'm just going through, just reading all those, the content. Okay, main things will come when we are going to discuss about the cyclins. That's still, different types of cyclins and their role is still left, okay? With that, the cell cycle gets over. Okay. So prophase 1, early prophase have got the 5 stages. It is the longest phase of the meiosis in which the pairing of homologous chromosomes will take place. The paired and the replicated chromosomes are called bivalents or the tetrads, which has 2 chromosomes and the 4 chromatids. Leptotene. So in this stage, the chromatin fiber uncoiling takes place and condensation of that chromosome gets started. Okay, each homologous pair starts to arrange in a lengthwise manners along the each other. So now what is happening? Nuclear envelope will start, slowly disappearing, and the chromosome starts to condense. Zygotene. In this phase, pairing of homologous chromosomes occurs, and here synapses happens. So synapses क्या होता है? जहाँ पे आपके homologous recombination is taking place. So one chromosome coming from mother and the another one coming from the father is there. So then here in between the 2 arms of the different non-sister chromatid, it is taking place. Okay. Pachytail. So here the crossing over is taking place. So the non-sister chromatids of the homologous chromosomes exchange their parts, and once the synapsis gets complete, the chromosome gets more condensed, and then they form a tetrad. Okay. Diplotene. So chiasmata formation takes place in this stage, which represents the site of crossing over. Okay. So here you can see, like, this is the area where the synaptonemal complex is formed and the chiasma, as you can see, it is formed and the exchange is happening. Okay. Now because of this exchange only, the recombinant of string is formed. Everyone knows about it, I believe. Then coming to the dikinesis. So this is the final stage of the prophase 1, and in this phase, the shortening and thickening of the paired chromosomes takes place. Nuclear memory is going to disappear, chromosome condenses further, and the condensation makes the four chromatids of the complex disappear in this phase, and now homologous chromosome movement is going to happen. Okay? So in the late prophase, microtubule formation will happen, centrosome gets duplicated. Okay, they will move to the opposite poles. Okay, so central which moves towards the opposite poles, and the asteroid formation from the central gets started, and thus at the end, okay, at the end of the prophase, the spindle apparatus are formed. Okay, now coming to the metaphase 1. So we have discussed in the meiosis about the different stages. Okay, so we discussed about the leptotin, zygotin, pachytin, diplotin, and the dikinesis. So all the 5 stages are coming under the prophase 1 of meiosis 1. Okay, in metaphase, the central reach the opposite poles, spindle apparatus are formed. They try to, with the head tensile method, they try to attach to the centromere of the chromosomes. Okay, now again, same thing. Here again, you have got the 3 types of the microtubule. That polar one, kinetochore, and the astral one. Same thing is repeated. Okay, guys. Now, anaphase 1, same. Homologous pair are getting splits and moves towards the opposite pole, and here it is your reductional division. ठीक है? तो पहले क्या था आपका 2n था, right? Now half year n n. Sorry, पहले इसको 4n किया, then 2n 2n, and then after that this is going to become n n n. Okay. Telophase 1. Each centriole represents a centrosome. Chromosome gets converted into chromatin. Okay, as you can see again, telophase 1 में क्या हो रहा है? Nuclear envelope आपका वापस आ रहा है. Chromosome जो है आपका chromatin में बन रहा है. Okay, and then finally cytokinesis will happen. This is again with the help of the actin and myosin 2. Now, interkinesis. What is interkinesis? It is the interface between the meiosis 1 and meiosis 2. Okay, but here nuclear material synthesis is not happening. So ये जो present है उसी को ही आप divide करते हो. So right now we have got the 2 cells. So after the first meiosis 1, we have got the 2 cells now. Okay, so again it has got the 2 centrosomes. Nuclear envelope is there, nucleus is there, chromatin is there. Now what is there? Meiosis 2. Meiosis 2 is nothing but just like your mitosis. Okay. So again, आपका centriole move होगा, right? Chromatin is going to condense, nuclear envelope is going to disappear. Again, metaphase 2, the chromosomes are aligned on a single plate, right? Spindle fibers are getting attached to the kinetochore over here, and they get aligned in the single. So यहाँ पे दो दिखाया because at the same time 2 daughter cells were present, right? So ये दोनों में दिखाया है. Anaphase 2, sister chromatids are getting separated now to the opposite poles. And at the telophase 2, they are getting separated. Again, the cleavage furrow is formed with the help of the actin and myosin 2, and followed by the cytokinesis. Okay, clear? So that was all about your, mitosis and the meiosis. So it was just, I was going through because I believe everyone understands what mitosis and meiosis 1 is. Now coming back to the regulation. Now the regulation, okay, cell division cycle regulation. Okay, so it is crucial to ensure proper segregation of genetic material. Otherwise, what will happen? No normal development will take place, or eventually the cell will form the cancer. So the key regulatory protein is your CDK, cyclin and the cyclin dependent kinases. And all these cyclin dependent kinases are your serine threonine protein kinases. So what does it mean? It means only on the serine or threonine OH group, the phosphate group will be attached. Okay, please remember they are your serine threonine protein kinases. Cyclin is a regulatory unit and CDK is your catalytic subunit. Same thing, right? जैसे हमने MPF में पढ़ा था. So we have got your cyclin B and CDK1, right? तो यहाँ पे ये कैटालिटिक हो गया, ये रेगुलेटरी हो गया ना. So they transfer the phosphate group. It causes a phosphorylation causes change in the enzymatic activity of the substrate to interact with the other proteins. So mainly, if you remember, I have said also that in the cell cycle regulation, mainly the phosphorylation is taking place. So either it is going to make it active or inactive. Okay. So CDKs, they are activated by binding to its regulatory units called the cyclins. So till now, 9 CDKs have been identified, of which 5 are active in the cell cycle. For example, during G1, we have got CDK 4 and 6, 2, S में CDK 2, and in G2M, we have got the CDK 1. Okay. So the cyclic CDK complex is formed as a result of the interaction between the cyclin and the CDK. So this conserved across cyclin, so the region of the CDK where the cyclin is binding is known as the stere. So this is the structure. Here you can see the cyclin A is a, CDK2 is a, and then they interact. Okay. So this region is known as the stere helix over here. Now cell cycle cyclins. This is also very, very important for you to understand the graph, because this is what comes for the exam. Okay. Let's go slowly and understand this. So this is the cyclin CDK complexes of the cell cycle control system. So the concentration of the 3 major cyclin types oscillate between the cell cycle. Okay. But हमने यहाँ पे CDK नहीं दिखाया है, सिर्फ cyclin का दिखाया है. So the cyclin which is present in the G1 S phase is known as the G1 S cyclin. S phase cyclin is S cyclin. और जो माइटोसिस के लिए चाहिए वो उसको बोलेंगे हम M cyclin. ठीक है. Now in the late G1, as you can see G1 आपका यहाँ से स्टार्ट हो रहा है, ये G1 है, राइट. दिस इज योर स्टार्ट रेस्ट्रिक्शन पॉइंट. इफ यू रिमेंबर, ये क्रॉस हो गया, देन ओनली it's a signal that the cell should enter the S phase. Remember the restriction point or the start point we have discussed, right? So once this is crossed, then only the cell enters to your S phase. Okay. So here we have got the increased concentration of your G1 and S cycle levels. So what will happen? This will result in the formation of your G1S CDK. Correct? So this formation will result in the activation of the proteins which will help in the progression from G1 to S. Clear? Itna clear hai? Now, at the start, as you can see, यहाँ से आपका S cyclin increase होना शुरू हो गया है, which is at your G1 phase. Correct? G1 S cyclin is making sure that the cell enters the S phase. At that same time, at the middle of that, like at the near G1, that is the start point, your S cyclin starts increasing. Now you have got the S-CDK cyclin complex ready, right? So iska kya kaam hoga? To start or initiate your DNA replication. Okay, this much is clear. The function of G1 S-CDK and S-CDK. Now, as you see throughout the S phase, S cyclin concentration is increased. Okay. Now, धीरे धीरे where the G2 is starting, there slowly the M cyclin concentration is increasing. Okay, so M cyclin CDK is formed towards the end of your G2. Okay, now what will happen? This is ensuring that the cell is ready for your mitosis. Did you get it? Okay, so G2 में क्या काम होता है? There is a checkpoint which ensures that there is no error happened during the DNA replication, या कोई damage तो नहीं है DNA को. So all that will be repaired in your G2, which eventually will result in the increased concentration of your M cyclin. Okay. Now M cyclin MCDK is formed. Now this will enter into your mitosis. There again you have got another checkpoint called APC/C. What is this? Anaphase promoting complex or cyclosome. This will again ensure that everything is proper, okay, and it will initiate the transition from metaphase to anaphase. And the chromosomes will be then separated. And again after that, at the end of the mitosis, G1 will start. Clear? Now let's see only the concentration of your cyclins. So in the G1, you have got high concentration of G1 and S cyclin. Correct? This is happening after crossing the start point. After crossing the start point. Then at the S phase, of course, it is the S cyclin. Okay. Now at the G2, there is a increasing concentration of your M cyclin. Clear? Then towards the M, जहां पे आपका APC आएगा, okay, जहां पे आपका metaphase to anaphase promotion हो रहा है, there the M cyclin concentration will start declining. Okay, as well as the concentration of S cyclin is declining at the here at the G2 phase, the concentration is declining of your S cyclin. In short, yeah. Clear? Guys, this is important. Okay, again and again I'm telling. Is it clear? The cell cycle cyclins? Okay, good. Same thing. Okay, same thing with respect to the CDKs we have given here. So if you see at the G1 phase, okay, we have got CDK4 and CDK6 along with the cyclin T. Correct. Restriction point आते आते आपका cyclin E activate हो जाता है along with the CDK2. So this is for your S phase. ठीक है. Now, once CDK2 cyclin E is there, it crosses the restriction point. It enters the S phase where CDK2 instead of E will bind to A. ठीक है. Now, it will progress. When it enters G2, there we have got CDK1 binding to your cyclin A and then B. Then it will allow the entry into the mitosis. Okay. Now let's talk about the cyclins in detail. So as we saw, cyclins are required at different stages of the cell cycle. Cyclin CDK complex are there D1, D2, D3, which all binds to the CDK4 and 6. It is necessary for the entry in the G1. E binds to 2 to regulate the progression from G1 to S phase. Cyclin A and 2 is required during the S phase. 2 and A, okay? तो ये अगर डायग्राम याद रहेगा तो बहुत easy हो जाएगा then. In late G2 and early mitosis, cyclin CDK1 promotes the entry into the M phase. Now it is with the B, as you see over here. Okay. CDK2 cyclin A यहाँ था. As you enter G2, A CDK1 के साथ है. Now for the mitosis, it is CDK1 with the cyclin B. That is your MPF, if you remember, maturation promoting factors. So far so good, guys. Able to remember? Is this figure is clear? ये समझ में आया? ये समझ में आया तो हम आगे क्या डिस्कस करने वाले वो और आसान हो जाएगा. Shall I proceed then? Or take a minute to remember this? अभी याद कर लो so that whatever we are going to discuss now will become clear to you. Okay, good. Krishita, is it clear?
Yes, ma'am.
Good. Okay, so cyclins are originally named because of their concentration. It increases and decreases in a cycle. So the oscillation of the cycles, namely fluctuations in the cyclin gene expression and the destruction by the ubiquitin mediated proteasome pathway induces oscillations in the CDK activity to derive the cell cycle. So why we are saying that oscillation in CDK activity? Because जैसे increase हो रहा है, so activity आपकी increase हो रही है. Then as it is progressing to the different stages of the cell cycle, these cyclins are also declining, right? So obviously the activity won't be present over there. So cyclins form complex with the CDK, gets activated, and its activation is only complete after the phosphorylation. Okay. So cyclin and CDK binds together. The cyclin themselves have no enzymatic activity but have binding sites for some substrates and target the CDK to specific cellular locations. So what are the types of the cyclins? G1S cyclins are in the late G1. Now why we say late G1? Because it is after the start point. Okay, please remember. And they fall in the early S phase. So that's why it is G1S. So this cyclin complex begins to induce the initial process of DNA replication by arresting the system that prevent the S-phase CDK activity in G1. Now this is the stage, okay guys, where the cell decides if all the required machinery for the DNA replication is available, then only these cyclins will bind to the CDK and activate. Okay. Now the cyclins promote other activities also to progress the cell cycle, such as centrosome duplication or spindle pole body in the case of the yeast. Okay, so the rise in presence of G1S cyclin is paralleled by rise in S cyclins. So it means the cell is progressing towards the S phase. G1 cyclins do not behave like other cyclins in that the concentration increases gradually throughout the cell cycle based on the cell growth and the external growth regulatory factors. The presence of G cyclins coordinates cell growth with the entry to a new cell cycle. So what we are saying here is that if you have growth factor, then the G1 is there entering into the cell cycle. Okay. If growth factor is not there, it will ensure that the G1 cyclins, okay, they get that signal and the concentration of G1 cyclin is less. So it enters your G0. Now, Kaposi sarcoma herpes virus, it encodes DA type cyclin which binds CDK6. Okay, and when it is binding, it is causing the cancers, Kaposi sarcoma cancer. So it's a virus. अब उसका cyclin आके हमारे CDK को bind करेगा. So obviously it is going to lead to the formation of the cancer. Guys, join back. Hardly 5 minutes are left. Let's complete this topic. Join back. Cyclin D. So now let's talk about cyclin D. So where cyclin D is present in the G1, it binds your CDK 4 and 6. Okay. So it is initiated during the G1. It derives your G1 to S phase transition when the growth factors and the mitogens are present. Okay. Then only the cell starts expressing your cyclin D. Now, कैसे होता है ये? Growth factors, it works through RTK. What is RTK? It is your receptor tyrosine kinase. So remember your tyrosine kinase, how it is working. So then this is activating your RAS-RAF-ERK pathway, which will result in the activation of your MYC protein. Now MYC will increase the rate of transcription of your cyclin D. Okay, and then it can go interact with the CDK4 and CDK6. In short, clear? So what is happening? The growth factor is there. It is coming and binding to the cell. Okay. Now it is binding to your RTK receptor tyrosine kinase. So it is activating the cytosolic domain over here. Autophosphorylation, all this thing is happening. Now what will happen then? It is going to activate your RAS protein. RAS के बाद RAF के बाद ERK, MERK pathway. So this will result in the activation of your MYC protein. Okay. Now this will get increased. This will enter your nucleus, act as a transcription factor, and there it is going to regulate the expression of your cyclin D. Clear, guys? Okay. Now, in the case of the yeast, it is known as CLN3. It is a homologue of cyclin D. It interacts with CDC28 during G1. In proliferating cells, because of continuous signal by the growth factor, cyclin D CDK forces complex starts accumulation. Now this is important because this will phosphorylate your RB, and the phosphorylation of RB is important for your S phase progression. Phosphorylation of RB causes dissociation of RB from the E to F, which is the transcription factor for cyclin E, and it is important for your S phase progression. So guys, did you get it? Like how from G1 to S phase transition is happening? We have earlier talked about RB कि वो कैसे काम करता है, ठीक है? So for that, it is your cyclin D and CDK4 which will activate your RB, ठीक है? So कैसे रहता है? If you remember, RB is bound to your E2F. This gets phosphorylated by cyclin D and CDK4/6 activity. So because of this phosphorylation, what will happen? RB will it will dissociate with the E2F. Now, E2F is a transcription factor which is required for your cyclin E. So जब RB आपका dissociate होगा E2F से, then only E2F is ready to act as transcription factor for the synthesis of cyclin E. Now, when the cyclin E concentration is increasing, then only it can enter the S phase, where it can bind to your other CDKs. Get it? Guys, able to follow? Shruti, is it clear? Yeah. Okay. So we know about the S cyclins then. So they bind to the CDK and forms a complex which helps in the gene replication. The level of S cyclins remains high, not only throughout the S phase, but through the G2 and early mitosis as well to promote the early events in the mitosis. Okay. Now coming to the cyclin E. So the transition from G1 to S phase requires binding between E and CDK2. Cyclin E and CDK2 complex will phosphorylate your KIP inhibitor of cyclin D. Remember, CYP and KIP inhibitors are there. So those are this. So cyclin E CDK2 is going to phosphorylate your KIP inhibitor of cyclin D. So cyclin D remains unbounded form as the cyclin E is already bounded with the CDK2. इससे क्या होगा? The cyclin D का जो काम था, to activate more of RB so that, more E2F is there and cyclin E is there. Now that is stopped. Okay. The phosphorylated form of P27-KIP1 is then tagged by ubiquitin and it is degraded by ubiquitin proteasome complex. Okay. Now this will allow or promote expression of cyclin A and it is going to progress to your S phase. Clear? Now coming to the M cyclins. So now the M cyclin concentration is increasing. It is entering the mitosis. Okay. So cell changes in the cell cycle like the assembly of mitotic spindles and alignment of the cystochromatids. This all is done by the M cyclin CDK complex. The destruction of M cyclins during the metaphase and anaphase after the spindle assembly checkpoint is satisfied, it is causing the exit of the mitosis and cytokinesis. So यहाँ पे क्या हुआ? Spindle assembly checkpoint क्या है? जो आपका anaphase में जो metaphase में जो आपका plan होता है कि like this kinetochore should kinetochore should be bound with these spindle fibers, right? So all when this is done, then आपका destruction of M cyclin will start. Okay, so expression of cyclins is all we understand with the help of the immunocytochemically, that's with the help of the antibodies ki kaun sa, at which stage this particular cell is present. So that you can all study by the immunocytochemically or with the help of the FACS flow cytometry. Now A and B, so this is present in your M phase, correct? So they play regulation of two different stages of the cell cycle. It can associate with 2 and 1. Association of A with 2 is required for passage into S phase, while the association of 1 is required for M phase. Okay. B is your mitotic cyclin. We have already discussed that along with the CDK1, it forms maturation promoting factor, or here otherwise known as mitosis promoting factor. Okay. So this is necessary for the progression of the cells into and out of the M phase of the cell cycle. So it is produced or it is synthesized in the S phase, but it is taken to the nucleus only at the last stage. Okay. So cyclin B or the MPF, what it does, so we have seen the function of MPF, nuclear envelope breakdown, then lamins ka phosphorylation, chromatin condensation, all this is happening because of the MPF. And at the late anaphase, there the cyclin B concentration will fall off. It means it's giving a signal that the cell needs to exit the mitosis. Same thing is given over here. See, at G1, the concentration is declining of your G1 and S cyclins. S phase CDKs, it is increasing and it is there till anaphase. This is same thing. जितना आपका S phase का cyclin रहेगा उतना आपका CDK रहेगा. And towards the G2, M cyclins will start. So it will form a complex with the mitotic cyclin dependent kinases, right? And then again it will fall. And at the end of the anaphase, the G1 cyclins will start increasing. Okay. Now same thing with respect to your CDKs as given over here. Same cycle. Okay. Almost same cycle. So cyclin A and B have destruction box, and cyclin D and E contains pest. What is pest? Proline, glutamic acid, serine, and threonine sequences, which is required for efficient ubiquitin mediated cyclin proteolysis at the end of the phase of the cell cycle. Now, ubiquitin mediated proteolysis of cyclin is catalyzed by ubiquitin ligase enzyme. So this all we have discussed, right? SKPF Cullen box, which is present, and APC is responsible for the ubiquitination and the destruction of the cyclins. So how we saw the synthesis. Okay, like how the A2F is helping in the synthesis or the gene expression of the other cyclins. Now, if suppose we talk about the destruction. Destruction के लिए, how it is happening? APC is activating your proteasome. Okay, and they have got these destruction box present. Destruction box मतलब जो ये pest domain है, it is present at which the ubiquitin comes and gives the tag. Okay, and once these cyclins have these tag of ubiquitin, it is meant for the destruction only. और ये कौन करता है आपका APC and the SCF. SCF क्या है? SKP1 Cullen F box. Okay, clear guys? This is the summary what we have just discussed, and we have talked about V1, CDC25, like how it is activating, regulating, right? We have also discussed about the INK4 family, which is an inhibitor, then CYP and KIP family, which is again an inhibitor. So this is just the summary. Okay, let's go through once. So CDK activity is regulated by the three mechanisms: synthesis and degradation of cyclins, or the phosphorylation or binding with the inhibitors. So these are the three ways by which the cyclin dependent kinases can be regulated. So phosphorylation, you have seen like V1 and MYTN1 proteins are there which will phosphorylate. Okay, tyrosine 15, threonine 14, threonine 161, that is the place where it is getting activated or inactivated by phosphorylation. Then we have got CDC25 phosphatase, which will dephosphorylate the size and will activate. Okay, अगर CDC25 inactivate नहीं होता है, then it remains inactive. Now, if suppose inhibitors are there, so if they go and bind to these CDKs, then also the activity can be regulated. For example, INK4 family is for your G1 CDK 4 and 6. CYP or KIP families inhibitors are for your CDK2 activity. So these are the three mechanisms by which CDK activity can be regulated. That's all guys for today. I'll just stop.